All right, I see Mark from San Francisco State. Welcome. Well, thank you again for your time and for joining OriGene's webinar on how to make and use lentivirus. Just a little housekeeping before we get into the presentation. Everyone here is muted. If you have any questions during the presentation, there's a tab for questions on the right-hand side. Go ahead and type the questions, and at the end of the presentation, we'll save some time and answer the questions. For those using smartphones, you should have a question box at the bottom of the screen. And so far, if you want to keep practicing with the question box, just go ahead and type in your current location. We also have slides that you can download, and that's at the handouts right below the question box. You can just click on there, and it should open up, or you can drag and drop on your desktop. All right, for those just joining us, thank you again and welcome. I'll be your host today. My name is Dr. Shoei Takeste, and I'm the lead scientist for lentiviral and AAV products here at OriGene. Viral vectors as a gene therapy tool, gene delivery tool is becoming increasingly more popular, and scientists want to experience the convenience, the efficiency, and ability to target difficult cells. It works great for both in vivo and in vitro sites. OriGene houses over 30,000 human clones ready to be shuttled into viral vectors. Think of your favorite gene. Chances are we already have a clone ready in our sites. We've been supplying lentivectors and particles to our customers, and we have a vast experience in this area. We recognize the growing demand to learn how to make lentiparticles, so today we want to share with you our knowledge and help you start on your next experiment with lentivirus. This presentation is geared for both new and experienced lentiviral users in the audience. If you're familiar with the introductions, please be patient as we have new updates on ways you can have high titer virus and increase your activity. Now, without further ado, I'll turn over the time to Dr. Louie, who is our product manager of lentiviral products. She obtained her PhD from University of Maryland, Baltimore, and went on to complete her postdoctoral training at Harvard Medical School before joining the company. Without further ado, Dr. Louie.
Thank you, Shui, for the introduction. Thank you, everyone, for joining the webinar. So today, I'm going to talk about the beauty of the lenti, the advantages, biosafety, and how to make lentivirus. So this is the overview. So the first is why use lenti. Then is it safe to use? And then we'll talk about what lenti products can OriGene offer. And then is how to make lentivirus. So what is lentivirus? Lenti means slow. So lentivirus is a slow replicating retrovirus. The research use lentivirus is derived from HIV-1. It's enveloped virus. The genome is a positive RNA, and it has structural genes and regulatory genes and accessory genes. So why use the lentivirus? Because there is a need to deliver genes into cells, especially the hard to transfect cells, the primary cells, and stem cells. The regular plasmid transfection fails to deliver genes into those cells. However, lentivirus can efficiently deliver genes into those cells. So the engineered lentivirus, the genome is mostly replaced by target gene, your gene of interest. So through infection, your gene of interest will be delivered into those cells. So this is a comparison. Why choose lentivirus over plasmid transfection? So for process, lentivirus is very simple. Just add to your cells. No need for transfection reagent. But with a plasmid, of course, you need to find an efficient transfection reagent. If it's possible, you're able to find one. Sometimes there's no good transfection reagent for your cells. So then delivery efficiency. lentivirus is very high for most cells. For plasmid transfection, it varies. Only a few common cell lines, the transfection efficiency is high. So talking about cell spectrum, so lentivirus infects most cell types, dividing, non-dividing, primary cells, easy to transfect, hard to transfect, can infect most cell types. For plasmid transfection, only limited cell types such as HEC293, HeLa, etc. And some cells, it is very difficult to transfect. You may need electroporation, then the cells will not be happy. And to make stable cells, the genome integration, lentivirus, the integration is very high. Plasmid, random integration into the genome is very low. So usually you have to do antibiotic selection. You still take a long time to make stable cells. For in vivo study, lentivirus can be used to infect tissues or inject in animals. But with the plasmid, there's no good transfection reagent transfecting tissues. So clearly, lentivirus is a more efficient and preferred way to deliver genes into cells or animals. So this is a short summary. Lentivirus is an efficient gene delivery tool because it has high transfection efficiency. It infects both dividing and non-dividing cells. It can stably integrate into the host genome. So this is to show you the transfection efficiency with the lentivirus. GFP lentivirus was used to transduce those hard to transfect cells like epithelial cells, mast cells, and smooth muscle cells. We can see almost 100% of the cells are green, which means they are transduced by the virus. So it's a very high transfection efficiency. So this slide is showing the in vivo use of the lentivirus. Lenti-GFP virus was injected into cell mice by blood vessel. And a week later, in liver and spleen, very nice GFP expression. Even 3 months later, because the lentivirus can stably integrate into the genome, so the GFP is still nicely expressed. So lentivirus is very simple and convenient. Just add to your cells, and the genes will be expressed, and you will get your results after 2 days. So I have to show you lenti is a great gene delivery tool. The question is it safe to use? Some of you may be concerned. And the concern is, can cells infected with the lentivirus produce more virus? The answer is no, because there's only one round of the infection illustrated here. So lentivirus infects your cells and integrate into the genome. The gene gets expressed. However, the viral genome is not replicated, so there's no more viruses produced. But mainly implemented in the lenti system, lenti-vector design. So the third generation lenti-vector is replication deficient. The first safety factor is splitting the essential replication elements into separate plasmids. So this is a lenti-vector. Some people call it a transfer vector. It mainly contains your gene of interest and the corresponding promoter, and then flanking with the LTRs, the packaging signal site. The other viral genes are provided on separate plasmids, supporting the genome viral replication and packaging. It's called here it's called a packaging plasmid. When the lenti-vector and the packaging plasmid co-transfect together into cells, and the packaging plasmid will provide the viral proteins to package this lenti-vector into pseudoparticles. Only the lenti-vector containing the LTRs and packaging signal is packaged into the viral particles. The packaging plasmid are not. So that's why it's very safe. Another safe factor is the packaging plasmid are on 3 separate plasmids. Makes it even safer. And another important safety factor is in the LTRs. The farm from the LTR is a hybrid. It does not need the viral transactivator tag anymore. And the 3' LTR is a thin, self-inactivating because the deletion in U3. So after integrating into the host genome, this viral genome cannot replicate because of the deletion in U3. So thin makes the virus to be replication deficient. So therefore, after infecting your target cells, no more virus is produced. However, so lentivirus, still a virus, precautions need to be followed. You will need to wear lab coat, gloves, work in tissue culture hood, the bleach liquid with alkali solid. Just basically, if you know how to work with the tissue culture, you know how to handle lentivirus. For the details, you can follow the NIH guideline showing here. And we also have a biosafety video if you want to know more details about the biosafety. It's posted on our Lenti page. Here is the URL to on YouTube. So next I will talk about what Lenti products does OriGene offer to help your research. The product offering is comprehensive. It's genome-wide, every gene, human, mouse, and rat. For gene overexpression is lenti-ORF, and for gene knockdown is lenti-shRNA. We also have a CRISPR vectors, the all-in-one with the gRNA and the Cas9 in one vector, or have the components separate, gRNA only or Cas9 only. I'll give you more details later. For the lenti-ORF and lenti-shRNA, the products are offered with the 2 formats, either the lenti-plasmid, of course, with the ORF already cloned, shRNA already cloned. You can make the virus in your own lab. Another format, you can get the lenti particle directly. It's ready to use, directly added to your cells. So this is showing the comparison between lenti-plasmid versus the viral particles. If you get lenti-plasmid, you need to get a packaging kit. That's the cost, and the time is about 1 week. To measure the titer, you need a titer kit, and this time is needed. So it's about 1 to 2 weeks to make it ready to use particles. If you get particles, so no more steps, just directly add to your cells. As I mentioned earlier, the 2 format lenti-ORF, just give you more details about lenti-ORF products. The lentiviral particles is 10 to the 7th. TU per ml, ready to use. Okay, so the lenti-ORF clones, OriGene's advantage is because we already have the high quality ORF clones. Those are already made in regular mammalian expressing vectors. So we only need to subclone the inserts into the lenti vector. So each gene, each variant is available in 4 different lenti vectors. This shows 2 Puro vector under P2A Puro. Either has a C-terminal fusion, make a DDK tag. DDK is the same as a flag. You can use the tag antibody to detect the protein expression or for protein purification. And this has a C-terminal fusion GFP tag, so it's a GFP tagged vector. So it can be used for subcellular localization. For these 2 vectors does not have Puro, so since the packaging there is a limit for lentivirus between LTRs, so without Puro, a larger insert can be cloned into the vector. OriGene also has over 30 lenti-cloning vectors. So either non-tagged or different tags or different selection markers, and the ORF can be easily cloned into those vectors by simple cutting the religate, and the gene will be in frame with those tags. So now talk about lenti-shRNA, still two formats available. The shRNA is a 29-mer, which is more potent. Also genome-wide antigen, human, mouse, and the rat. The product is offered as a kit. So it's 4 target-specific shRNAs and 1 scramble control, which means that if you get the lentiviral particles, you will receive 5 different viruses. And we also offer performance guarantee of at least 1 out of the 4 will give you 70% knockdown. So this is the lenti-shRNA vector. So between the LTRs, which will be packaged into the lentivirus, there are 3 major functional elements: shRNA under your 6 promoter, and pyrimidine under SV40, and GFP under CMV. So therefore, the transduced cells or infected cells will express your target shRNA. It will resist to pure selection, and the cells will be green. So you can use the GFP to enrich the transduced cells if it's needed. So this shows CRISPR lenti vectors. This is all-in-one. It has cloning site to clone the gRNA sequence here. So gRNA will be under U6 promoter and also has Cas9 under CMV promoter. So after infecting cells with this virus, the RNA will bring Cas9 to the genome specific location and lead to double-stranded break. And this is a gRNA-only vector. Here is a puro replace the Cas9, so there's no Cas9 in this vector, and this is a Cas9-only vector. So sometimes the researchers need to make a Cas9 stable cell so they can use Cas9 only, and then just introduce the gRNA-only vector. So for some researchers, they may need a high titer of virus for hard to transduce cells or in vivo studies. We offer custom lentivirus service. The titer is from 10 to the 7th to 10 to the 10th TU per ml. We can offer larger volume too. And the custom lenti production can be provided using OriGene's lenti vector or customers or your lenti vector. Custom quotes can be requested online. You will clearly see on the website under the service or under lenti page 2, you will see the link. So next I will present you some lenti validation data. This is the ORF clone in the GFP tax vector and 72 hours post-transduction in hexagonal T cells. You can see great transduction efficiency and very high expression level too. And this gene is a transmembrane protein, and you can see the membrane localization. So suggesting that the GFP tagged protein is localized to the right cellular compartment. So this is showing the lenti-ORF of different sizes, ranging from 0.7 to 6.5 kb. And this is the Western blot with a tag antibody, and you can see the expression is very nice. So this data showing gene expression using different MOI. MOI is a multiplicity of infection. That means the number of copy of virus per cell. This is Western blot with a tag antibody. You can see the higher the MOI, the more virus per cell, the higher gene expression. So sometimes you may need to use different expression levels for your study. You can use a different MOI, different amount of virus to infect your cells. So this is the data showing the lenti-shRNA. This is scramble control. As I showed earlier, the lenti shRNA vector has GFP, so they are all green, showing nice expression. And this is shRNA targeting the GFP in the vector, and so significant knockdown of the GFP. So next, I will talk about how to make lentivirus. So there are a few steps to make the lentivirus. First, to package the virus, co-transfecting your lenti vector together with the packaging plasmid into HEX-NET3 cells. And then the titer, to measure the titer, to find out the exact titer, you may need to do MOI optimization. If the titer is low, you may need to increase the titer, virus concentration, to concentrate the virus. Next is the stability issue. Lentivirus is known to lose infectivity during storage, even at -80. So you may need to, in the buffer, increase the stability. So for every step, OriGene has a kit or reagent that can be used. So for the packaging kit, we have a very good high packaging efficiency kit and a titer kit to measure the titer. The concentrator is a reagent can be used to concentrate the virus. And the lenti stabilizer is the buffer that lentivirus can be resuspended into which can increase the stability during storage. I will give you more details in the next few slides. So this is how to package the virus. So either your lenti vector containing ORF, shRNA, or CRISPR co-transfect the lenti vector together with the packaging plasmid into HEX-NET3 T cells, and then 2 days later, the virus will be produced in a supernatant. So this is our lenti VPAC lenti packaging kit. So it contains 2 vials. 1 vial of lyophilized lenti packaging mixed together at the optimal ratio, and 1 vial of a transfection reagent. So the packaging mix is at the optimal ratio to make sure the high packaging efficiency. Also, the step in transfection, that's critical for the packaging efficiency too. So that's why we include the turbofactin, which is a very good transfection reagent that will ensure the packaging efficiency too. And this packaging kit is for 3rd generation lenti vector packaging. So the protocol is on the website. It's basically just a code transfection. It has how many cells to see all the detailed protocol associated with the kit. So to measure the titer, you can use the P24 ELISA kit. P24 is the viral capsid. There are about 2,000 molecules per virus. So you can convert the P24 amount to the particle numbers. So this is a diagram to show how the ELISA kit works. P24 capture antibody is coated on a plate. Then you add the viral samples, contains P24, and then add the P24 detection antibody. You will measure the P24 amount. This is our titer kit. It's a 1-wash titer kit because it has a simplified workflow, only 1 wash before the substrate. The conventional kit contains 3 more washes in between. It's also convenient, only room temperature incubation needed, so no more 37 incubation. And the shortened protocol, especially in the sample binding step, our kit only needs 2 hours, the conventional needs overnight. So it's a convenient and shortened protocol. So to increase viral titer using the lenti concentrator, that's a reagent can be added to the viral supernatant to concentrate the virus. This is before concentration. This is after the concentration. Dramatic difference. You may be curious why before concentration the signal, the fluorescence is so low. That's because we want to tell the difference before and after. We use a shorter exposure time. So when using the regular exposure time, the before concentration is also very bright, just like the image here. So using this lenti concentrator, you can rescue low titer, using it up to 100-fold. And the protocol is very simple. It's a mix and a centrifuge, no ultracentrifuge needed. It's just a regular centrifugation. And there's an additional benefit. The concentrated virus can be protected from freeze and thaws. So frequent freeze and thaws, it does not affect the infectivity. So this is the lenti stabilizer, which can stabilize the virus during storage. This is a fresh GFP virus. And then when incubated at 37 for 5 days, which is equal to -80 for 1 year, in the typical production lentivirus media, I see the virus almost lost the infectivity. When the virus stored in the lenti stabilizer, the infectivity is well preserved. It's almost the same as the fresh virus after 1 year. So all the lentiviral particles provided from OriGene are in the lenti stabilizer buffer. So it's very stable during storage. We also, if you want more details, we also have a lentivirus production video that's posted on our lenti page. Here is the URL. So actually, we have more lenti resources on our website. So like the lenti protocol, the packaging and transaction protocol, how to optimize MOI, and if lentivirus work with the neurons, FAQ. And this is the video as mentioned earlier, how to make lentivirus. And this is recorded webinar, how to make lentivirus, with more detailed step-by-step protocols. This is a lenti webinar is recorded. That's a general lenti webinar. This is about 50 video I mentioned earlier. So you can refer your friends, or you can go to those lenti resources and learn about the lentivirus. So to summarize, lenti system is a very powerful gene delivery tool. It can infect most cell types, in vitro and in vivo. So OriGene's genome-wide lenti product, lenti-ORF, SHR, and CRISPR, which can help your research. Also, we offer the packaging kit, the titer kit, and lenti concentrator, and the stabilizer to help your lenti production, making the lentivirus. I will be happy to answer any questions.
Thank you, Dr. Louie. We'll go ahead and take some time for questions. And just a reminder for those that joined us late, be sure to type your questions into the question box on the control panel on your right. We'll do our best to answer all or most of the questions. If we do run out of time, we'll send an email with answers to everyone. So we'll go forward with the first question here. What is the mechanism of 3' LTR self-inactivation?
Okay, that's a good question and a complicated question too. That's during viral replication after integrating into the genome. That's a little complicated to explain because the deletion in the 3' U3 region, it cannot transcribe the viral genome.
Correct. And just to add a little bit on that is when the virus infects the cell, it has to go through a reverse transcription step. So the sequence on the 3' LTR will be transferred to the 5' LTR. So once your gene of interest is already integrated, now you have both LTRs that are important for replication of the gene of the virus are both inactivated, and that's why they call it the self-inactivating system. We have another question. Could you compare lenti to retrovirus?
Okay, yes, very good. I had that in my other recorded webinar. For virus, first, it only infects dividing cells. So that's for a lot of primary, non-dividing primary cells, you cannot use retrovirus. And another thing is that the retrovirus titer is a lot lower than lentivirus. So sometimes you make stable cell take a long time because the titer is low, the transduction efficiency is low, so it takes a lot longer time to make stable cells. So that's the two major differences between retrovirus and lentivirus.
Okay. How can I determine my titer for transducing NK92 cells?
You probably want to know what MOI you want to use. Okay, so for that you need to optimize it. You can use a control particle, like we have the control particle expressing the GFP. So you can use a different titer, different MOI to see which MOI can affect your cells better.
And to add again on that, if you ordered a viral particle, I would recommend to titrate it in a 96-well plate. A lot of scientists tend to do it in a 24-well plate, but you end up using a lot of your virus. So just doing a small 96-well plate, different volume or concentration of the virus with a fixed amount of cell, and just go with the one that shows a good transducing efficiency. So we're getting a lot of questions about stabilizer. One is asking how long does the stabilizer last and the catalog number. The catalog number can be found on our site.
Yeah, it's a TR30038.
TR30038.
Yes, I mean, in our hand, in our test, it can definitely stabilize the virus for 1 year. We haven't tested longer yet.
Yeah.
So after 1 year, it still preserves the infectivity very well.
And they're asking if we have a question about if they can buy the stabilizer reagent as a standalone product.
Okay, yes, that can be ordered separately because you need to change the buffer. So on the web page is that it's probably better to use together with if you have other buffer changing mechanism, that's okay too. So you can use our concentrator, just spin down the virus and then just change the buffer into the stabilizer.
Yeah. We have a question that's related to in vivo efficiency of knocking down target genes in implanted tumor. How can we validate the knocking down efficiency?
The knocking down efficiency, you could isolate the tumor out, I guess.
Yeah. At first, I think even before proceeding to in vivo, you'd want to do some in vitro analysis to make sure that the sequence that you're using is actually being able to knock down your gene of interest. And then from there, you can move on to, you'll be confident enough to move into an in vivo aspect of the research. And again, like Dr. Louis suggested, you might need to exercise out the tumor and do a Western blot or qPCR depending on the amount that you have. Okay. So really great questions. Keep them coming. Do you have any recommended transduction methods? Reverse transduction, spinification, AAC. I can answer that. With the reagent that we use, turbofectin, and I've used a lot of other different reagents in the market, you get really good transfection efficiency. But when you get to the transduction method, that depends on your cell line, and it's a trial and error. What works for one cell line might not work for a different cell line, and sometimes we do have other additions. So with polybrin is a common one, but some cells are toxic when you add polybrin or a different concentration. So playing around with the different kinds, and at the end of the day is making sure that you have the correct amount of virus per cell that you're trying to transduce that will increase the efficiency.
Yes, for that, I also recommend using the control particle because the cost is lower too, and it has fluorescence, so you can monitor the transduction efficiency.
That's true.
Yeah.
We have a question. For the do-it-yourself kit, how does one introduce the gene of interest into the lentivector prior to co-transfection with the packaging construct?
Oh, okay, so you basically how to clone the ORF into the lentivector. So, I mean, you can get the insert, or you can directly get the lenti-ORF plasmid from OriGene. We have a genome-wide offering, human mouse rat. If you have the insert in your lab, you need to clone into a lentivector just according to the multiple cloning sites, just clone into the vector.
Yeah, you'll have to, it's going to be a lot of cloning, and we're known for being efficient with cloning, so that if it's hard to do it yourself, we can always talk on that. Rose is asking, I want to knock down my gene of interest in specific tissues, such as adipose tissue in mouse. Can I use the lenti-shRNA, and how is the mode of delivery to specifically target adipose tissue?
Adipose tissue. So at least first, as Shui mentioned earlier, at least test the shRNA sequences to see if it can knock down your gene in cell lines. Make sure that then I believe it's injection, right?
Yeah, so that would be, you have different methods of injection, so I don't know what would work best. We're not mouse, but
right. But definitely,
injection,
yeah,
we'll get it there.
For in vivo study, you will need high titer too, so you can get high titer lentivirus.
And for high titer virus, what we do here is we use ultracentrifuge to concentrate it down, so you get a really nice pure viral production for in vivo studies. Does lentivirus show any cell type specificity? If so, what are those cell types, and what determines its cell type specificity? Any hard to infect cells by lentivirus according to experience?
Yes, for that, it's not specific to cell types, because the envelope is a VSVG, basically infects any cells. For some cells, it's maybe more difficult to transduce. You just there's the MOI chart on our website for some common cell lines. You can take a look, or you can optimize the titrate out your cell line to see which MOI to use. Definitely different cell lines use different MOI.
All right. If you have any more questions, go ahead and type them in. So far, it looks like we've answered most of the questions that I see here. All right, someone is asking about cell passaging of the cells. We had a similar question earlier, the quality of the cells.
Yeah, you can.
So we normally use 293Ts. It's always good to make sure that for lentis, you're using cell lines that have not passaged over 10. You know, if your cells are happy and they're adhering well, then that will result into a good titer after transfection. It'll give you a good transfection efficiency. So always use not over 10. What is the different promoters can add to ORF and vectors? So I guess the question is asking what different promoters do we have in our vectors?
Okay, yes. For our lenti-vector current promoter, we have CMV promoter and the EF1A promoter. And we already made neuron-specific promoters now. It's under test now, so it should be available in the next month or so. So if you need a different promoter, you can contact us. We can make it for you.
All right. It looks like we've covered most of the questions. If I've missed anything, we'll follow up with an email answering most of these questions again. Thank you so much, and we appreciate you being here. We'd love to hear from you. We have a great team of scientists ready to help you for your specific needs and experimental details. Give us a call, and we can customize your orders from cloning to viral particles and deliver within a competitive time frame. Thanks again for joining us today, and we'll see you next time.
Thank you. Bye
Bye.
now.